scientists like Don Potts, collaborators in Mexico, because most of the field work we're doing is down in Mexico in the coral reefs, offshore from Cancun, and colleagues from Woods Hole, and of course funding agencies that supported this research. So just an outline of what I'm going to cover today. I'm going to discuss what is ocean acidification and how it works, and kind of the most important aspect of it is why do we care about it, why should we care about it, and then I'm going to discuss some overall research directions that the community, not necessarily my lab, has been taking in order to address these issues of ocean acidification, and I'll spend after this introduction, most of the time discussing a specific venue of research that I'm conducting with my lab down in Mexico that focuses on coral reefs and how ocean acidification may or may not impact coral reefs. So just to jump right into the background and introduction, I assume most of you in this room have seen this figure at some point in your classes, but in the lectures you're coming to or heard in the news. This is a figure that shows the concentration of carbon dioxide collected every month at the top of the mountain in Hawaii and measured since the 1960s. It's called the Keeling curve, named after Charles Keeling, who started this program. And as you can see, while there's wiggles up and down, which show seasonal changes dictated by vegetation in the northern hemisphere, during winter there's less vegetation, so there's more carbon dioxide in the atmosphere. In the summer and spring, there's more vegetation in the northern hemisphere. The plants absorb or take some of the CO2, so we see these wiggles. But this increase over time is primarily due to anthropogenic fossil fuel burning and deforestation, which resulted in emissions of CO2 into the atmosphere and accumulation in the atmosphere. This is a very well-known issue that a lot of debate, political, scientific, is ongoing for the past few centuries about, because carbon dioxide is a greenhouse gas and it's one of the major causes for climate change and global warming, with implications you can see here: sea level rise, this is the temperature rise, sea level rise, and so on. The most important aspect about this is not so much that atmospheric CO2 is rising in high, but the rate at which it's increasing. As you can see here, and I don't expect you to see the small fonts, but this shows more longer ice core records, so glacial, interglacial, over the past 400,000 years. There's been changes in atmospheric CO2, but this is where we are today. And in the geological record, it has probably been as high as it is today, or maybe even higher. But the rate, this sharp rate of increase, is probably never been seen before, at least for the past 50 million years. So we're in a very strange time in terms of geological history. But this is all well known. Another thing, though, that the CO2 in the atmosphere does is it doesn't stay there. The Earth functions, it has a lot of feedbacks and equilibration. So if you look at that previous curve, the fossil fuel curve, this blue line is this line over here, just smooth it. Oh, sorry, this one is what we measure in Hawaii, but we know how much fossil fuel we're actually burning. We know how many barrels of oil are being used, and we can calculate what should be there in the atmosphere. So this blue line is what, based on our fuel consumption, should have been in the atmosphere. As you can see, what actually is there is less than what should be there. So where is this 30% of CO2 going? Well, it's going in the oceans. It's also going into regrowth of vegetation, but a lot of it is going into the ocean. There is no question or debate about that. That is a simple law of physical chemistry. Any gases that we have in the atmosphere equilibrate with the water they interact with. The equilibration and the amount depends on the concentration in the atmosphere and the temperature of the fluid it equilibrates with. So all of this 30% of this missing CO2 is absorbed in the ocean. And the question is, what is it doing? How is it impacting the ocean? Honestly, for many years, people were aware of that, but did not really think that it may have any major impact. We were all focused on global climate change and global warming and sea level rise, and we somewhat, even though we knew about it, ignored the impact of this CO2. And this CO2 absorption into the ocean is also called the other CO2 problem, to differentiate it from the global warming problem, or ocean acidification. What happens is, as the CO2 dissolves in seawater, it changes the chemistry of the ocean such that the oceans become more acidic. They are not acidic. The oceans are still basic, but they're tending towards more lower pH or acidic. I'm going to stop right here for a second to show you what I mean or how this works. So can I have 4 volunteers? How about the kids in the room, or the younger folks? And there's 4 of you exactly. Why don't you come over here? What I'm going to do is, Chris gave me some seawater, and we're going to give you a little bit of seawater over here. Does anybody know what the pH of seawater is about? It varies from place to place, but it's around 8. On average, it's around 8. Actually, here off California, it's a little bit less than 8. Okay. How about you join us too? I know you're older, but that's fine. So, and this is a pH indicator, and around 8 or 8.2, 8.4, it has a reddish color. I'm going to put this over here, and you can see that it has a red pinkish tint to it. So, and we can, if anybody does not believe me, I've got pH sticks. You can stick it in there and read the reading. So, what happens when we exhale? What do we breathe out? We breathe out CO2. So let's see what happens to this water when we breathe CO2 into it. This pH indicator loses its red color and becomes yellow as the pH drops. And let's see how fast this happens while I explain what's going on in the ocean. You can go stand wherever you want, and don't hide the glass or the cup as you're blowing. Just blow in bubbles and we'll see what happens. Okay, meanwhile, I'll see what's going on here. So this is a little demo that shows the carbon dioxide going into the ocean. As it goes into the ocean, the CO2 reacts with water to form carbonic acid. That's what lowers the pH. Now, this is not only what happens. As you have more carbonic acid, you can see this green stuff over here shows that the carbonic acid increases. It dissociates to bicarbonate, and this H plus here, the protons, are what's changing the ocean's pH. Now, the protons also react with carbonate ions, so the concentration of carbonate ion decreases, the protons increase, causing acidity, and these other 2 forms also increase. So the end result of this CO2 dissolving into the ocean is that you have lower pH or more protons and less carbonate. Okay? The change, you can show people around. It's yellow. So this is basically what happens. It's pretty simple. CO2 goes into the water, and you can leave these here and we go back to seed, and the ocean becomes more acidic. So how do we know that it's happening? Actually, there's lots of monitoring programs that are pretty clearly showing that CO2 goes into the ocean. All of this red, yellow, and light blue shows how much anthropogenic CO2 is coming into the ocean. And we know it's anthropogenic because we can measure the isotopes, the carbon isotopes of these CO2, which are different than the oceanic ones. The CO2 that we're burning from fossil fuel has distinct signatures, and we can follow the fact that this CO2 in these depths, up to about 1,500 meters in the ocean already, comes from fossil fuel burning. We can see that as this Mauna Loa CO2 record in the atmosphere increases, you can see that the pH of the ocean decreases, and the CO2, or the partial pressure of CO2 dissolved in seawater that's in the open ocean, in the middle of the Pacific off Hawaii, is increasing. So the atmosphere is increasing, the CO2 in the ocean is increasing, and pH is decreasing. So there's a lot of evidence. There's no controversial or no questions about that. The evidence are clear. So if we go ahead and actually determine how much the pH changed, so this map here shows you the change in pH, or the delta, which means the seawater pH before we started burning fossil fuel to what it is now, between 1700 and 1990, and white is zero change. All the colors show changes towards more negative pH. And there's not a single place in the ocean that has not become more negative between 1700 and 1990, with the larger changes at the high latitudes, as you can see here. Now this looks pretty small, doesn't it? 0.1, 0.2. You have to keep in mind that pH is measured on a log scale. So a 0.1 pH unit is 30% change. That's pretty big. So this is not tiny numbers over here. These are pretty impressive changes in the pH in the ocean. If we look at it in a different perspective, how does the pH decrease more from a modeling perspective from 1875 and projecting into the future? So the blues are pH is 8.2, 8.3, 1875, you can see that much of the ocean was above pH 8. You can see some lighter blue. These are natural variability where deep water comes up to the surface through a process called upwelling. The deep water has lower pH naturally because of respiration, and that you can see that in areas of upwelling are slightly lower pH, but overall everything is above 8. 1995, at higher latitudes, we're seeing more of these light blue. Now, 2050, the projection is that most of the ocean will be below 8, between 8 and 7.9, and if we go to 2100, everything is below 7.8 or lower. These are big changes in the ocean's pH. Now, this is based on modeling, based on predictions of how we will keep on behaving, how much fossil fuel will keep on burning, what the vegetation response will, how much will dissolve. So there is some variability there in the absolute values, but the trend is clear. Even if we stop using fossil fuel today, go all green, use only solar, wind, alternative energy, and we stop burning fossil fuels, the amount of CO2 that is currently in the atmosphere will equilibrate with the ocean, resulting in these kind of scenarios. So unless we pump all of the air in the atmosphere through filters and clean it up, which we're not going to do, the trajectory is clear. So what are the potential impacts to marine organisms and ecosystems? And there's a long list over here, and I'm stressing potential because we don't actually know. This is a hot area of research. This is what a huge number of scientists in the community are now trying to figure out. Now that we, the coin kind of it dawned on us that this is happening, it's like, okay, what is it that we can do about it? What can we think might happen? So there's going to be a reduction in calcification, and I'm going to talk a little bit more about that shortly. There's going to be shifts in nutrients availability, and trace metals, which will impact the base of the food chain because the phytoplankton, on which all of the fish and further on in the food chain rely on, need nutrients and trace metals to grow. There's going to be shifts in the phytoplankton diversity, growth, potentially reduction in growth, production, and lifespan of some adult organisms and larvae, and a whole list of physiological and ecological impact. There's going to be changes also in the biogeography because each organism has their ideal niche of where they're best adapted to survive, and that's going to changes in food web. There's also going to be physical changes as well. Sound absorption is going to change because of these changes in chemistry. They impact mammals that need to communicate, homing ability, which depend on the chemistry of the water, and so on. So there's a lot of potential impacts. And because of that, the community was somewhat alert and interested in doing something about it. So what can we do? And a whole bunch of federal and national and international agencies, abbreviated down here, sat together and said, okay, what can we tell the community? What can we recommend the scientists to do about that? So they recommended a fully integrated program of laboratory, mesocosms, field monitoring, modeling approaches that will all take place in order to provide policymakers with the information that they can design management strategies that will lower the impact that we can do, or will enable us to adapt. Because if we can't do anything about it, how can we adapt to it such that it doesn't impact our fisheries, our ecosystems, tourism, whatever the benefits or the services that the oceans provide to us. And it could be anywhere from tourism, shipping, security and safety, Navy issues, and mostly fisheries and shellfish industry as well. So this is the recommendation, and as I said, a lot of people started working on that, monitoring trends by developing sensitive sensors and deploying them, calibrations of these sensors. We're looking at organisms and ecosystems responses by taking these organisms and growing them in the lab or monitoring them in the field under various control conditions to see how they react and how they adapt. We're doing a lot of modeling work to predict the future. And then there's also more interaction with the social scientists about adaptation strategies, emission reduction, sequestration, and so on. And of course, education, kind of explaining to the general public, and particularly K through 12 and high school kids, that this is happening so they are aware of the issues. This is a graph that I put together that shows the number of papers published on ocean acidification, and it looks like the CO2 curve. So you can see until like the 1990s, hardly anybody did any work on that because it didn't raise to the awareness. Nobody even thought it's going to be an issue. And then since 2000, bang, everybody is working because some of it is motivated by the fact that the funding agencies give money to the scientists to do this work, and they're interested in that. Okay, so now to focus a little bit on some of the work that is related to ocean acidification, and that's calcification. If you remember when I showed the solution of CO2, one of the impact was that carbonate ion concentrations in the ocean are going to decrease. The carbonate ions will react with the protons that are released from the acid, and there will be less carbonate in the ocean. Who needs carbonates in the ocean? Shells? Anything that produces shells? Other things? Some phytoplankton need carbonates. Corals. So anything that produces carbonate shells, aragonite or calcite. So I've got some shell rubble here, and I can give that to you if you want to move around. And I've got some dilute acid. And if I put the dilute acid in, you can't see it from here, but I will move it around, or if somebody wants to come and tell you, basically it's fizzing. It's fizzing because the carbonate is dissolving with the acid. If you take carbonate and you put your bottle of Coke on it, it will disappear. The doctors are not lying about our teeth, which are not carbonate, are dissolving. So yeah, if I take the shells and I add acid to them, they're going to dissolve away. Now this is much more acidic than the ocean will be. But the fact that the organism, so it's not necessarily that they will dissolve away. However, the fact that they don't have enough carbonate out there will make it much harder physiologically for these organisms to produce their carbonate shells. And it may impact their larvae, which are less protected. There was a couple of years ago, a big news item about shellfish off Oregon, where they were growing shellfish for consumption, and the industry completely crashed because they were pumping waters from offshore into their tanks. The water were low pH, none of the spat grew, and they had a year where the industry crashed. They now realize that, and with very simple bubbling or equilibration of the water, or just timing when they're pumping the water and monitoring, they can prevent that. But that's one consequence that may happen. So just to show you how this looks very similar to the pH map, but this is now the change in carbonate ion availability. And again, you see everything here is colorful, meaning there is a decrease by these many millimoles per meter cube of water from 1700 to 1990, and you can see pretty much most of the ocean has been impacted and has less carbonate ions. And these carbonate ions, as I said, are the skeleton parts of all of these shells, seashells, clams, mussels. Some fish have otholates that are from carbonates, and these are internal, so they may be less sensitive, but a lot of different phyla, different organisms are all producing carbonates. These are the phytoplankton over here, the coccolithophores, the pteropods, which are the food base for krill, for fish up in the high latitudes. Yes?
Would ostracods be one of them?
Ostracods will be impacted as well. They're agonitic, and they would go.
Why is the Pacific Ocean more affected than?
That's the Atlantic, actually.
Oh, I'm sorry.
So why is it? That's a good question. So the reason is that that's where the water is forming. So this is at a depth horizon that is below the surface, and because deep water is forming, or water is penetrating the depth of the ocean in the North Atlantic, it's difficult. Most of the water that flows into the deeper ocean originates up here, and some of it down in the Southern Ocean, and it flows. So this water is just younger, and it has more of this imprint in it. Over here, this younger, more CO2-rich water has not quite arrived yet.
You also got pink in the Bay of Bengal.
Yep. And that could be the major reason for that is the temperature. Solubility is temperature dependent. The circulation there is a little bit more constrained. So if you have warmer waters, they may dissolve more CO2 in them. So it's a balance between circulation and solubility and upwelling. And that's partly what controls the distribution. But overall, it's going to be low everywhere. So back to our organisms, one of the organisms that most likely will be impacted are corals. And corals are rich ecosystems. They provide important services. They're nurseries for fishing. They're very important for tourism. They have historical and cultural benefits. They protect the seashores from wave storms because there's this reef that blocks the waves from coming. So they have a lot of reasons for us to worry about them and be concerned about them. But the most important part is they make their shells from a carbonate mineral called aragonite that is very soluble. And they're not very well isolated from the environment. They're a very thin layer of tissue that separates the organisms from the environment, so it's not like some internal organ inside a big fish or a mammal or something like that. So they're exposed, and because they're also impacted by a lot of other potential problems like bleaching and global warming and overfishing and people snorkeling and trampling on them and impacting them, so they might already be sensitive. So if we look at where corals are, all of these dots over here are distribution of corals, and you can see the scale here shows the aragonite saturation, and that actually tells us how much carbonate and how much calcium is in the water for them to produce their shells. And they're abundant where the aragonite saturation is high, 4 or 5 supersaturated, and that's the optimal range for calcification for corals. And you can see in 1765, all of the corals pretty much were in areas where, or the shallow corals were in areas where it was optimal for them to grow in terms of calcifying or producing their shells. If you look at what it is in 95, the red areas shrank, although most of the high coral population are still within the optimal range. But if you look at the Caribbean right here, they're going into the less than optimal, the adequate range. And if we project to 2100, nothing's going to be optimal. The highest populations here are going to be in the marginal range at best. Yes?
How do you get numbers from 1765? And do you have anything from like 1900 to show progress before?
So this is based on a model, not on actual measurements of what the CO2 in the atmosphere was at these times based on ice core records, and then assuming the solubility of the CO2 in the atmosphere, you can calculate the saturation level. And then you can get numbers from 1765 and so on. And you can do that for every year. They just, in order to show the changes, they skip that. So it's based on whatever data we do have, plus what we know about CO2 in the atmosphere, and all of the future one is what models project CO2 in the atmosphere would be. Does that make sense? Was another question? Yes.
Are these levels available? Like, I mean, I teach high school, so if I wanted my students to research this and look it up, is that something that they can get these values?
Absolutely. And if you want, after the talk, I can give you web pages where you can download either raw data or maps like that, and a lot of other information. So there's several web pages run by NOAA and by Ethica, which is the EU Ocean Acidification Program, where they put tons of papers, databases, and so on. We actually have Monterey, Embari down in Moss Landing. They monitor the pH in Monterey Bay, and they have that data, both the pH and the CO2 available as well if you want something more local. Okay, so it doesn't look very promising. And indeed, over the last few years, you can see these headlines both in the newspapers and in scientific journals. Growing acidity may kill corals, increasing ocean acidification, eroding corals, and so on. Marine threats, acidification hits the barrier reef. It's really, the coral reefs are such an important and, that close to many of us, even if we don't go diving. I think all the kids saw with the Nemo movie and can imagine that coral reefs are important and they don't want Nemo to disappear either, because Nemos depend on the coral reefs to grow. Okay, so what do we do? Again, this is the projection model. These are the predicted responses. I think I explained this pretty much, what is being suggested. It's pretty much the prediction is the doom for the corals. They won't be there. Well, is that so? That's a very good question, and Bob Garrison is here, and he did a lot of geological research on coral reefs, and the corals were around for many, many, many millions of years. There were periods where they were more abundant or less abundant, but they disappeared for a while and then they show up again. So it's not clear, and these periods may have had much higher CO2 than we have today. So it's not clear how is that. There seems to be some kind of contradiction between their ability to survive in low pH and the fact that we see them continuously in the geological record with different levels of abundances. So this was a nice work that Mo Stein and Danny Chernow published in 2007, and they're showing that if they took this coral from the Red Sea and they let it grow at pH levels that are predicted for 2100, just with running seawater, that they bubbled CO2 in, and what they found is that the corals, or the skeleton of the corals, disappear. But the corals themselves didn't die. They were pretty happy, but they did not form skeletons. So that may be good news for corals, and that may explain how these corals kind of held on in the geological record when the pH of the ocean may have been low, and then they showed up again and started calcifying again when the conditions changed. However, this is bad news for coral reefs, because the corals themselves would look like anemones. They would not build a reef. So all of the ecosystem that lives and strives from the actual framework of the reef, all these nooks and crannies and places to hide and so on, and the protection from the waves may disappear. Now, of course, we don't know that that is what will happen, but that may be one thing. If we take these corals and just grow them, that's how a coral skeleton looks like today, and then we subject them to different levels of CO2, you can see that they're dissolving. It looks like there's somewhat of a threshold that after a saturation level of about 2.5, they just kind of decrease and disappear in terms of their skeletons. If we do the same thing we just put an in situ bubble, make a little cage and bubble CO2, you can see the impacts of the ocean acidification in the field where you have a larger another coral in a beaker, and the results are the same. Generally speaking, the calcification rates decrease as the aragonite saturation decreases as well, and that's a compilation of lots of studies, but not all studies show that. Some studies, in fact, show that if you take normal CO2 or high CO2 at pH at 25 degrees, there's no difference. But if you increase the temperature, then they don't do very well. So there's also all sorts of confounding multiple parameters that may impact the corals that we need to try understanding. Again, if you look at the linear extension rate, which is how many millimeters per year the coral grows, it looks like they've changed a lot, but generally there's a decrease in growth in some studies, but other studies show that over the past, from 1700 to the present, there's variability, but there isn't a clear decline. Of course, if you go to nature and look at corals, it's hard to differentiate different stressors. In the lab, you can control the conditions. In nature, multiple things happen at the same time. So it's not really clear what's going on. Yes?
What is the blue curve on that top graph?
Over here, this is the pH, and this is the delta boron. And you can see that there's been natural fluctuations in pH that's an area in the Great Barrier Reef. And the pH was changing back and forth, but there isn't, like you'd expect from the 1700s to 2000, a general decrease in pH. And we see fluctuations, but not a unidirectional decrease over here. Now, this is not measurements that were taken in the water. These were measurements that were reconstructed from corals. They took a coral core and they counted the layers to reconstruct the time, and then they measured the boron isotopes in this carbonate. The boron isotopes is a proxy or recorder of environmental pH. And based on the boron isotopes, this is the data, they reconstructed the pH. And they didn't see a clear decline. So it's not really clear, and there's a lot more we need to do in order to study. What seems to be clear is coralline algae, which are algae that cement the corals in the coral reefs, do seem to disappear. This is a beaker with low pH, and this with a regular pH, and this is low pH, and you can see that fleshy green algae grow, and the coralline algae, these darker pinkish things, disappear. And they're important for the reef structure. Okay, so key questions that remain are is there a chance for corals to acclimate and adapt? If you expose them to these conditions, will certain coral species make it and others disappear? Do they have the genetic ability to actually make up or compensate for the lack of carbonate? And in order to address that, we can't really grow the corals in a beaker, because we need to look at several generations in their natural environment in order to address that and see if there are some adaptations. To get to an adaptation, you can't keep something in a beaker for 3 days or 3 weeks, when the lifespan of a coral could be, as a colony, could be hundreds of years. So we need to kind of do that in the field. Yes, back there.
I'm sorry, I don't quite understand that model for oceanic pH. Is that questioning the earlier model you showed for?
No, it's just saying that it's more complicated than we think. There's no question that the pH in the ocean is decreasing globally, but locally, in an ecosystem, there's other local parameters that impact the pH. And the pH in the coral, where the coral grows its shell, may be very different than it is outside. So it's possible that even though the external pH is low and there's less carbonate in the environment, under certain conditions, corals can compensate for that and maintain inside their tissue, inside their living cells in the area where they're calcifying enough high pH in order to continue building their shells or their structures.
How closely is the pH related to the carbonate?
Very. They're all interdependent. That if one changes, the other changes in a very predictable way. Okay, so to go out and look at in the field, there's a few places where you can actually go and look at coral ecosystems that have been exposed to low pH conditions over a long time span. And that's one paper that was published in 2011 of Papua New Guinea. There's these volcanic vents. CO2 is spewing from volcanic activity at the bottom of the ocean, close to the reef. And what they found is that at pH 8.1, there's lots of organisms and calcifying organisms. And near the vents, there aren't that many. But look at these. There are some corals that are there. Not as many, but if you look over here, these are all of the negative impacts. All of these things look worse, but there's three things that are actually better when you compare the vents to the non-vents, and these are the non-calcareous algae and the seagrass cover, which is what we'd expect. These are grasses that need CO2 for photosynthesis, but also this massive porrites corals. There's more of them close to the vents. So somehow they're able to survive that. And now we're going back to my specific work. My field site is in Mexico, off Cancun. This is the map of the Yucatan Peninsula in Mexico. And this area is a karstic environment. If you heard about all these caves that people go hiking in and so on, so rainwater falls, it penetrates into the caves, and it, through these caves and caverns, reaches the ocean where it discharges in springs. Now, because this water is rainwater, and it passes through the soil where there's a lot of bacterial respiration and CO2 is produced from the roots, then this water that discharges has high CO2 and low pH. Luckily, it's not fresh water because there is saltwater intrusion, so before the water discharges in these springs, it mixes with seawater, and the pH is just slightly lower than normal seawater pH. But it's still saturated with CO2, and you can see over here the water spewing out of little crevices in the rock, and you can see there's several submarine springs like that. Locally, they're called ojos or eyes because they're very focused or localized, and over, up and down, just about 500 meters offshore at the depths of 6 meters or so. And that's how they look underneath. There's like a little ledge, the water is spewing over here, and there's a nice reef growing all around it. If you look from the top, you can see this bubble over here. That's the impression of the water coming up because it is slightly less saline. You can see it raises up to the surface, and you can see it over here on a column vein. So what we did, we put some sensors to measure the pH in these springs, and outside the springs you can see there's lots of variability. This is expected. There is periods of high tide and low tide which will impact the discharge of the groundwater. There's rainy season and not rainy season, and it's a lagoon where there's a lot of seagrass, so during the day there's photosynthesis, and at night there's respiration. All of these parameters will impact the actual pH that we measure, but overall you can see that for the most of the time the pH is pretty low. It's below 7.6. There's a few peaks over here and there, but it's fairly low. Yes?
What's the x-axis then?
That's just days. It counts the days in the year. So each number here is a day. You can see there's variability, day and night variability, and so on. Sorry about that. Yeah. And these sensors were there for months and months at a time. And that's just a snapshot. always below ambient. Ambient pH there is 8.1. We also measure the calcium concentrations, the dissolved CO2, the alkalinity, all sorts of parameters that may impact the saturation level, and they're monitored continuously, and we calculate saturation levels. So the first thing we wanted to answer is, are there corals there? Which species of corals? How do they compare to areas without the springs? To do that, we did surveys. We just put little squares and we counted all of the organisms around there, the cover, the species, the numbers, along gradients of pH. And that's a very complicated figure, but it shows you up here the saturation level. So red is good, blue is where you have a spring and it's below saturation. You can see on short scales, these are meters over here, there's a lot of variability. But then right here, it's blue, it's undersaturated, and there's a coral. And over here again, you can see the spring over here, and there's a coral, and so on. So there are certainly corals living in these springs under undersaturated conditions. Presumably, good news, because there are some corals that can survive, and these are very low pHs. They're undersaturated, 7.6, and very worse than any predictions for the future. So that's actually pretty good. But if you do a little bit more digging, you can see that in the control environments where the saturation is about 2.5, you find about 10 species of coral. At the springs, there's only 3 species of corals. These corals tend to be not the ones that build the reef. They do calcify, they can grow, but they're not major reef-building corals. The coral area and the number of individual corals also is declining. So while this is good news that some corals can make it, it's really doesn't mean that there is not going to be a negative impact. Because if these corals can't build reefs, then we're not in such a good shape after all. But it's a system where we can learn about the mechanisms, the physiological mechanisms. What is different about these corals that help them survive in these extreme conditions? We can protect that area because these corals are already adapted. So if we want to keep corals that will stay there for the long term, we may want to protect that area so in the future we can use that as a bed for reintroducing corals to other environments. The fact that they don't make up the reefs is because other species of corals grow faster. But if they're not there, these may grow enough to form the reefs. Okay, the other thing we did is we drilled the corals and took cores in order to look at the growth rate of these corals. And these are the CT scans of corals that are from the spring and from outside the spring. And you can see that they grow if you measure centimeters per year, they grow at a similar rate. However, in this year where they grew, they put in much less material, much less carbonate, so their density is not as strong. And this blue here shows lower density. So while the extension rate is similar, except it's slightly lower here, the density drops dramatically, and calcification is the growth rate times the density. That's the amount that is being precipitated or formed, and that decreases. So what does that tell us? Yes, they grow, but they grow less dense. And because they're less dense, what it means is that boring organisms can attack them easier. So this is a core of the control versus the core in the low pH, and you can see that it's all eaten up. There's a big, as saturation decreases, the volume of board versus the volume of coral increases dramatically. So they're not as a strong framework. So again, they can grow, but they're not doing as well. In terms of their physiological health, their tissue thickness is the same, so they are healthy, they are growing, but they're putting less carbonate per unit time compared to the others. Now, all of these other things here, the symbionts and the nutrient levels, these are parameters that have been hypothesized to provide energy to corals to allow them to calcify at low pH. And I don't have data for that yet, but these are the directions we're studying now to see if maybe they have different symbionts, if there's more nutrients, if the symbiont density is higher, and that maybe is the mechanism that allows them to be able to calcify. The other thing that we're doing is we're doing transplantation experiments where we took corals from outside the springs and brought them into the springs, and corals of the same species, and then corals from the springs outside the springs, and we're letting them grow there. They're all marked. We're letting them grow there for a year, and then we'll go and look at their genetics to see what are the genetical makeup that maybe allows them to acclimate to these conditions. So hopefully at this time next year, we'll have all the genetic information that will tell us what are the genetics of these specific corals. And then we can know if other corals have this genetic makeup. We can target the genes of other corals, and if they have these genes that will allow them to overcome this low pH, then maybe we can again have some hope that they have the physiology and the genetic makeup that will allow them to overcome, assuming we provide them with some other conditions that will give them the energy to still grow and be able to overcome other hurdles, whether it is overfishing or higher temperatures or people trampling on them and so on. So with that, I'll open it for questions. Yes?
So this has been all about the acid, but what about the artificial reefs? You know, people are like dropping in cement structures or something. Is there a way to drop in more calcium to counter the acidification?
So, not really, because the ocean has lots and lots of calcium. The calcium is not a problem. They don't have the carbonate, and they need both. Now, if you add more calcium, yes, you can bring the saturation a little bit higher. But they actually are not limited by the calcium, they're limited by the carbonate. When they put the cement, it's not so much for the calcium, it's for substrate. They need substrate, they need hard substrate in order to, when they're pollen or when they're at their larval stage, they need to then settle onto a hard substrate, and that's where they put in that. But if we know, again, if we know that certain corals can do better, we can increase the substrate in these areas where these corals naturally live. We can protect them and make them refugia for stocks for future coral reefs. We can take them into aquariums and keep them there and then introduce them slowly till they get their genes and their physiology activated so they can be reintroduced. Corals are pretty hardy. You can take a piece of coral and trim it or take a branch of it and keep it alive in an aquarium, and if you feed it well at very low pHs and then it grows, gets acclimated, you can reintroduce it and it will grow in nature.
How prolific are they in reproducing? Are they like jellyfish where they can...
They tend to spawn, and then after a larvae stage, the larvae settles. The success rate is very low, but they spawn many, many gametes, so there's lots of them out there. And they tend to not reproduce very often. Most of them have one day a year, or a couple of days a year where they spawn, and then it's a big spawning event, and all the fish come to eat the baby corals. But that's their strategy. So their strategy is produce a lot of larvae, few of them survive, but then those that survive, once they settle and grow, usually they make it. And their different corals have different frequencies of reproducing that way. But they also can, if you cut a piece, you can then transplant it and it will grow because it's a colony. So that's kind of the spawning part makes the genetic variability, but they can also just grow from one colony without the sexual reproduction.
So we have time for about one more. Any last questions?
There were at one time some pessimistic predictions about the Great Barrier Reef. Is that still all true?
So the pessimistic predictions are still there, but I think we're learning that nature probably is more resilient if we protect it. So if we make an effort to not harm the corals by exposing them to multiple stressors. So if there's heating and low pH and overfishing and grazing and people, using dynamites to bomb them, they're not going to make it. But if we protect them and we make sure that they're not harmed in other ways, they might be able to overcome this one stressor at a time. And we can also be a little bit more proactive and not only protect them in the environment, but keep gene lines of corals that may make it better so we can reintroduce them into the environment potentially.
Okay, before we end things, I want to take a minute to thank our 4 acidifiers here.
Yes.
Spending yet another Science Sunday in the Seymour Center. So thank you guys so much. Hopefully we'll see you next time.